Hi, I'm Freya. I study biology, chemistry and maths A-levels. I'm going to be taking you through proteins for the A-level biology EdXLB course. So we're going to start by looking at amino acids. Proteins are made up of long chains of amino acids. The basic structure of amino acids consists of an amino group, a carboxyl group, and a variable R group. There are about 20 different amino acids that occur naturally. The R group determines which amino acid it is and affects the way it bonds with others. Amino acids join together by peptide bonds during a condensation reaction. This condensation reaction produces a molecule of water and a dipeptide. The dipeptide can join to more amino acids to form a single chain called a polypeptide. The highlighted area here is a peptide bond. Proteins have different levels of structure: primary, secondary, tertiary, and quaternary. The protein structure determines its properties and therefore function. For example, enzymes are globular proteins that have a very specific shape due to their structure. This is important to their function, as only one substrate will fit to its active site, therefore they will only catalyze one reaction. Primary structure is the sequence of amino acids forming the polypeptide. The amino acids are held together by peptide bonds. In this diagram, all the circles represent amino acids and the lines in between represent peptide bonds. Next, we have secondary structure. The secondary structure describes the way the peptide chain is coiled as a result of the hydrogen bonds between the carboxyl and amine groups. They can be beta-pleated sheet or an alpha helix shape. The tertiary structure describes the further folding of the chain into a globular 3D shape. It is held together by hydrogen bonds, ionic bonds, and disulfide bonds. These occur between the R groups of amino acids. This is an example of what it can look like. In the second diagram, you can see the bonding. Quaternary structure describes proteins with two or more chains of polypeptides linked together by hydrogen, ionic, and disulfide bonding. This is an example of a protein with quaternary structure, haemoglobin. So that completes the 4 levels of structure of proteins. Next, we're going to be comparing fibrous and globular proteins. Fibrous proteins have straight chains, whereas globular proteins are compact and often spherical. Fibrous proteins are insoluble, while globular proteins can be slightly soluble. Fibrous proteins are involved in structure, while globular proteins are involved in catalyst reactions as enzymes. You can see visually here the difference. So we're now going to look more in depth to a globular protein with the example of haemoglobin. Haemoglobin is found in red blood cells and is responsible for the transport of oxygen. It is a globular protein with 4 polypeptide chains, 2 alpha and 2 beta. It has 4 heme prosthetic groups. The heme part means it contains iron. This allows it to bind to oxygen. The fact it has 4 enables it to bind to oxygen 4 times more. Next, we're going to look at another example, which is collagen. Collagen provides strength to body tissues like tendons and skin. It is a fibrous protein with 3 polypeptide chains, 2 alpha-1 and 1 alpha-2. 3 helices of collagen are held together by lots of hydrogen bonds. The bonding is very tight because the third amino acid is glycine, which is very small, allowing it to fit into the small space of the triple helix. Proline and hydroxyproline surround glycine. They have big R groups that stay out of each other's way, which maintains the strong and insoluble structure of collagen. Collagen molecules can cross-link with one another to provide further strength as well. So finally, we're going to look at an example question. This example question links proteins to enzymes. An enzyme is a protein and has primary structure. Explain the meaning of the term primary structure. So this is 2 marks, so you need 2 points. You can either pause it and write down your answer, or you can just think about it. So here I'm going to show you the mark scheme for the question. You need the specific word sequence or order of amino acids, and you need the bonding, which is peptide bonds. Here we're going to look at another example question, which is bigger, using the same diagram as before, and your own knowledge of enzymes. Explain the importance of primary structure of an enzyme to its function. Again, you can pause it if you would like and write down your answer, or you can just think about it. This is the mark scheme. There are 10 points you can write about. I'm just going to pick out 5 example points that you could include in your answer, like mentioning that it's 3-dimensional folding and that the types of amino acids determine types of bonds, so these bonds would be hydrogen disulfide. Linking it to enzymes, you could say the position of the amino acids determines the shape of the active site. For a further point, you could say the active site has to be the correct shape to bind to the substrate. You could also mention the fact that it's globular or soluble. So with the end of that question, that sums up proteins for the A-level biology course. I hope this helped.